Hi, good afternoon. So this is a summary of the exam 3 results, the main version, which 250 out of 300 students took this. So that's a main one I want to analyze. As you can see here, this has a nice normal distribution of the performance overall, if you look at that. So which is good. But more than that, what I am pleased with these numbers, this number may look kind of a very similar to others. This 66%. Don't worry about it. The number is going to go up because this is rated based on 62 points. So the 2 point will be taken off and things are going to look much better. But more importantly, I see a lot of students shifted towards a higher grade, and I see less in this range. So that's another improvement. The third improvement I would say is this is a difficult material, biochemistry, metabolism and respiration, photosynthesis, concept-wise, content-wise, detail-wise, chemistry-wise, in many ways, this is difficult. So overall, the class did better than the other exam. Okay, now I'm going to go through each question individually, and then we can look at it as to how you approach these questions and so on. So I have mentioned about this before. Something I look at is a discrimination index. This shows as to, this provides a metric of how well a single question can tell the difference between students who do well and those who don't. So that's the main idea of this one. Anything less than 0.25 is not a good question. Then I reconsider, improve it in the next exam. Or if there's any additional points to be given, I consider that. So this is a very simple, concept understanding question. Energy coupling is the main topic of this question. How can this exergonic reaction powers the endergonic reaction? They are coupled. So the use of ATP in Calvin cycle, I didn't want to be very specific. I just said photosynthesis. And so that is the best answer. 77% of the student answered it correctly. But if you look at other example, ATP synthesis is endergonic, protein synthesis is endergonic, ATP hydrolysis is exergonic, simple diffusion is also exergonic. You don't need ATP there. Simple diffusion is exergonic and active transport is endergonic. And that one, 19 students selected that. In this case, you had to think about what type of active transport is possible. And simple diffusion is, again, without going through the protein, is going through the membrane or from point A to point B, so that also won't work. And active transport is endergonic. This is exergonic, so that's incorrect. This is a conceptual understanding question. The second law of thermodynamics states that every energy transfer and transformation results in increased randomness in the universe. So I state that very clearly. So it's already, the answer is given in the question. The living organism consume energy and release energy. This transfer and transformation in living organism doesn't increase randomness of the living organism, but the randomness does happen in the universe. What is the universe of that organism? Is in their surroundings. That's where the randomness happens. They are not exceptions to the law. They cannot continue to transform forever without being disorganized. The randomness is happening within the cells. We don't notice that. All the CO2, everything being released is within the cell and the water. Then we exhale it at the end. So that's the correct answer. Almost for 60% got it correct. And it's a very good discrimination index of 0.41, which is nice. This is also a conceptual understanding question. If a reaction is already spontaneous, exergonic, you don't need ATP hydrolysis. Okay, so I'm asking, Which one needs ATP hydrolysis is only an endergonic reaction like this with a positive delta G that is less than 7.3. I'm not even giving you any numbers here, but 60% students answered correctly. Exergonic reaction is a spontaneous reaction. It's already favorable reaction. It's happening on its own. You don't need to invest ATP to do that. So that is the conceptual understanding you need to have on this question, okay? The next question is, again, a graph interpretation. So I am giving you some graph you have never seen before. So learn how to interpret this figures, like understand what is x-axis. This is fructose 6-phosphate is a substrate given in a millimolar concentration. What is the y-axis? It's a velocity or the phosphofructokinase activity. And then you look at the response. So this is no inhibitors. There's a low ATP that acts like a stimulator. And this is just ATP and some AMP. ATP is more, and then there's some amount of AMP that is kind of a slow, but 1 millimolar ATP is kind of, slows down, acts like an allosteric enzyme. So this is what is the main thing that you want to look at. The sigmoidal shape of this is what indicates it's an allosteric enzyme. This also at a 0.31 discrimination index, and many of you said, and the one I'm asking is, what is the nature of this enzyme? What is the role of ATP in this reaction? So if you see this phosphofructokinase, it's a key regulatory enzyme in glycolysis, and ATP is an inhibitor, not an activator. So that is where the connection you need to understand and remember this type of question. So paying attention to detail will help you get the right answer, okay? So this is a recall. Either you know it or you don't know it. And we discussed this in a class question about ethanol and methanol poisoning. It also a very high discrimination index of 0.49. And accidental drinking of methanol can make one person blind or die. But the person who drank methanol by mistake is given ethanol as an antidote. How does it work? So in this case, I tell ADH is an enzyme that can convert both ethanol and methanol to respective product, acetaldehyde and formaldehyde. But acetaldehyde can be converted to a non-toxic molecule. It's like either you know this or you don't, or you got to kind of come up with an idea. So in this particular case, and ADH prefers ethanol, that is why it will not deal with methanol. And methanol is not an activator and ethanol is an inhibitor. That doesn't work. Both are substrate for this enzyme. It prefers one substrate over the other. So that is where the understanding comes, where many of you missed that question. A significant number, not many, 56 answered correctly. So the next question is, which of the following mechanisms are used to regulate enzyme activity? And this is select all that apply question. Many of you were very quick to select activators, inhibitors, feedback regulation, but some of you forgot about chemical modification, which includes phosphorylation. You have seen that in ATP, not a use of ATP in sodium potassium pump, proton pump. And this phosphorylation cascade in signal transduction, all that is also included, but this irreversible binding is done, enzyme is done. So you don't use that as a regulatory mechanism. So that is the main thing that you want to understand from this is recall and some understanding of the mechanism. Okay. So the next question, which outputs of glycolysis enters mitochondria and used for sugar formation? Where is direct recall? You know it, yeah, or you don't know it. So 88% got it correct. That's good. Glad you remembered. Also very high discrimination index. And select all that describe the process of respiration. And here I had repeated exergonic process twice. I meant to have endergonic in one of them, but I had repeated that, but I have given credit to all of you who selected that. But it is not an anabolic process. So that's the one thing that didn't get any credit. It is a breakdown process. It's a catabolic process. So that is the one where some of you missed the point. And 30% answered correctly, which is high for a select all that apply. Normally you get about 10%, 12%, or 7%, but this is very good. And understand the equation and answer the following question. The reaction of NAD plus becoming NADH is considered as a gaining hydrogen, is a reduction. And NAD plus is removing hydrogen from some other molecule like the food XH2 or something, RH2, and that is acting as an oxidizing agent. So this is understanding what the term oxidizing agent, reducing agent to mean. Oxidizing agent makes somebody else lose hydrogen or electron. Reducing agent makes somebody else gain hydrogen or electron. So in this case, it made it lose. So it's an oxidizing agent. Very high discrimination index, so I'm overall pleased with this exam results. Is a discriminated people who know and people who don't know. So the Krebs cycle makes only 2 ATP per glucose. This is the same question as we had in the square cap session. Most of the energy is used in the oxidation of acetyl-CoA, is used to reduce NAD plus and FAD to make NADH and FADH2. So that is 63% got it correct. And some of you got, again, confusion between oxidation reduction. Concept is very, very important. Oxidation is loss, reduction is gain. We talked about it, oil rig. And this is referring to electron and hydrogen. Why reduction is gain of electron? Electron is negative, so your positive charges go down. That is why it is called reduction. And oxidation is gain of oxygen. Just like phosphorylation is gain of phosphate, oxidation is gain of oxygen or loss of electron or hydrogen. And next question is same as I have given in a sample exam. Creatine phosphate is used as a supplement during intense workout. So when you're doing this is a substrate level phosphorylation. Kinase is the enzyme that does it. And ATP synthase comes in oxidative phosphorylation, substrate level phosphorylation. That's where ATP synthase comes. So you have to understand the mechanism, also remember some key facts. So you cannot, just because it is making ATP doesn't mean it's an ATP synthase. It's a kinase. Creatine phosphate kinase, or CPK, is the enzyme that would take the phosphate from creatine phosphate, give it to ADP to make ATP. This is like a quick burst of ATP synthesis. When you're doing workout and you're running out of oxygen, this mechanism helps you make ATP for a short while. Good discrimination index. And then why does oxidative? This is a pure comprehension, understanding. Compared to substrate level phosphorylation. This is a new question I made up. And so oxidative phosphorylation utilizes all the NADH and FADH2 generated in glycolysis, acetyl-CoA formation, and Krebs cycle to make a lot of ATP. So many of you did select that. Very good discrimination index. And some of you thought oxidative phosphorylation is oxidation, and reduction happened in substrate level phosphorylation. This is just to confuse you that both a process, substrate level and oxidative use oxidation process for removing electron, and that's how it happens. And conceptual understanding. The main purpose of alcohol fermentation is to regenerate NAD plus. That's the reason to keep it going. If you don't have NAD plus, okay, everything will come to a grinding halt. So you need to understand that purpose of that. You need to close that cycle. Pyruvate, CO2, pyruvate is an intermediate. Alcohol is a byproduct. CO2 is a byproduct. NADH need to be converted to NAD plus. That is where understanding them, when I am going through these things in the class, you need to pay attention to that. That's the reason, we have the lecture and I record it, I share with you. So you have so many resources, but it does take time to go through them, listen to them, re-listen to them, until you fully understand. Then you go to TAs and UGTAs or PLAs, office hours, learn from them. And so that is the deep learning is what they expect, not just memorizing blah, 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 blah. That's not going to help you, go to med school or pharmacy school or nursing school or whatever school you're dreaming to go to. Understand the material, connect the dots, see the big picture. And I have said that multiple times. And you need to see that connection between glycolysis and oxidative phosphorylation and back to glycolysis. This is where that particular question is. High discrimination index. This is also very good. What is exergonic process that directly drives endergonic process of ATP synthesis? And it's a chemiosmosis, and proton motive force is what drives that ATP synthesis and oxidative phosphorylation. The proton come back from high to low concentration through ATP synthase. It just acts like a turbine to rotate that ATP synthase to release that ATP so it can make more ATP. Okay, another conceptual understanding. So very important understanding of the mechanism. It's key to know how we make ATP. That's what keeps us alive. Study with that curiosity. And once you understand, you remember, it's going to stay for a long time. Okay, so that is what you need to focus on. Same in the coming exam. I'm covering a lot of experiments. You don't understand why the scientists did this. What does the results mean? How they proved their hypothesis and disproved the alternate hypothesis. It's a lot of understanding. This is not just reading some blah blah on a flashcard, memorizing this, memorizing that. It's like a laundry list memorization is not for a long term. Okay? You do need to know information, absolutely. That's your foundation knowledge, but you need to go beyond remembering information, understanding and connecting the dots. That's key for answering questions like this. So the next one is very similar to the fermentation question, even though this is a question we asked before in the square cap session. I ask you why oxygen is not an input in glycolysis, acetyl-CoA, or Krebs cycle, but everything will stop if oxygen is not present in oxidative phosphorylation, because oxygen is not there to accept the electrons from the electron transport chain coming from NADH and FADH2, and they become NAD plus and FAD. They need to go back into those reactions. You need to know that connection. Only half the class got it right. And it's a very high discrimination index. And so this is what is the purpose of electron shuttle? This is another thing that you need to understand. NADH is too large to get into mitochondria. They use smaller molecules like glycerol 3-phosphate or malate aspartate to give the electrons to them and come and move it into mitochondria for oxidative phosphorylation, because oxidative phosphorylation happens in mitochondria. So that is again an understanding of the material. It's very similar to that. 138 respondents. I'm sure this is the same 136 respondents got it. And this one has a little bit more. More people understood that. And so this is what many of you, the electrons are transferred from NAD plus to NADH. That's not the purpose of electron shuttle in the respiration process. And this is happening in oxidative phosphorylation, acetyl-CoA formation. So whenever I explain the why part, you want to pay attention and write it down. Because I say that subtle way, but you need to know that connection. That's the reason I say that. And next question, photorespiration occurs during hot and dry weather. During this time, plants must conserve water by closing their stomach. This is straight recall. And stroma is the liquid part of chloroplast inside the inner membrane. Don't confuse that with stoma. Spelling is important. And so whenever you also study these structures, you should be able to visualize them, just like seeing a dream. or an image. And many of you read fiction, paperback, and many of those fiction, they don't have any drawings at all. But you can imagine and visualize somebody doing this, somebody doing that. There's a magic castle somewhere. There's an unicorn living in an emerald forest or whatever. You study, you bring that to a visualization. That's same in biology or chemistry or psychology. Anything that you learn is including engineering. You need to learn to visualize in a three-dimensional way. That would help you remember, oh, that is leaf, this is epidermis, this is lower epidermis, that's a stomach, they are pores, the air goes in and things go in. So that will help you not make such mistakes. Like 22% of the class, 55 students missed it. So that is another thing, pay attention to detail. Okay, the next one. Even though light is not an input of Kelvin-Benson cycle, CAMPLAN fix CO2 in the nighttime by PEP carboxylase and wait until daytime to perform Kelvin-Benson cycle, because this is similar to the question about algae grown in light transferred to the dark. They stop reducing, phosphoglyceric acid into the glyceraldehyde 3-phosphate. I asked similar question in a different way. And I have told this also during my lecture, this Kelvin cycle always happens during daytime, even though light is not needed because it does need the output coming from the light reaction. Many of you did get that correct. But this one, I just give this, I just made this question to distract you. These are called distractors. So the respiration happens in the nighttime also and the daytime, but they don't need this oxygen. They can use the oxygen that's coming from the stomates. So that is where you kind of misunderstand if you don't remember the connection between these two pathways. Okay, key. Which of the following are inputs? Many of you did correct. This is the best select all that apply response I ever got so far. Okay, good, you remembered these things. And you have the same question here. Look at this. Kelvin-Benson cycle, you know that needs ATP and NADPH. And this question is asking the same. Look at that. So here, 66% got it correct. Here, 76% is not bad. It's not bad. So I think it's good you're making that connection. So that's not much different there. The next one is C4 and CAMPLANs, also 47% answered correct, which is also good. And I'm sure you're able to eliminate and you're getting better at the select all that apply. And so here I will go through, we are done with the multiple choice question. I'm going through the free response question. And after I'm done with this, I'll open it up and you can ask questions on any particular of them, one of them. And two exergonic process in the Kelvin-Benson cycle that results in an endergonic process to happen and include 3 chemicals involved in this. Going through the multiple choice questions will help. What do I mean by that? Kelvin-Benson cycle, it's ATP hydrolysis, NADPH oxidation to become NADP plus. These two are the exergonic reaction. CO2 becoming CH2O is an endergonic reaction. So the ATP hydrolysis powers the endergonic reaction. NADPH oxidation powers the reduction of CO2 to carbohydrate. That is the connection. I'm just giving you a hint. And then the next one is straight from discussion section. Okay. And, this is, again, 1/4 of the class kind of remembered it. Even if you don't remember anything from discussion section, I'm giving you a hint. Based on the figure legends and schematics, identify which compound is the enzyme. And I give you the answer. Dark blue is the enzyme. Then ask which one is the inhibitor. Okay. So, if you read this, non-competitive compound C. Okay, this is again a non-competitive inhibition. And it's also given there in this paragraph. And then if you go into the cofactor, and you know what are the cofactors are in this one, NADP, NADPH, organic cofactors. So that is something that you want to remember. And the cortisone and cortisol, like these things, the triangle you find, becoming this and this becoming that, and all that is very, you need to understand that's an active site, one is binding to that, and this is a recall and comprehension and understanding and interpretation. So whenever you see these questions in discussion section, I have given one in today's DSQ 11. So if you don't understand that chart or the figure in the discussion, you want to keep nagging the TAs and UGTAs and mentors until you fully understand and you can explain it to someone. Okay, that's a level of understanding I expect. So reduction oxidation, right, it happens in respiration. And I said focus on the electron carriers or organic cofactors. So if you remember NADH or NADPH, in this respiration is NADH and FADH2. So NAD becoming NADH, FAD becoming FADH2 is that reduction process. And acetyl-CoA becoming CO2, losing hydrogen is an oxidation process. Glucose becoming pyruvate is an oxidation process. So there are multiple examples. So here you have more people. If you look at that, only 27 in the bottom. And this one also not bad, 16% at the bottom. And comparing oxidative phosphorylation and photophosphorylation with 2 similarities and 2 differences, a lot of similarities, a lot of differences, and 82% got it, correct. And this is a straight recall question. But compare and contrast. So you should be able to compare, both make ATP, both use ADP-PI, both use ATP synthase, both use chemiosmosis. But differences will be oxidative phosphorylation mitochondrion, photophosphorylation in the chloroplast. Oxidative phosphorylation uses oxidation energy, photophosphorylation uses light energy. There are different ways that you can compare these, and and one happens in all organisms, all eukaryotes, or all organisms. And photophosphorylation occurs in only photosynthetic organisms. And there are so many things that you can compare and answer. Briefly describe how the structure of mitochondrion is so important for its function, include the parts and processes inside the mitochondrion. We talked about this also. That's to concentrate protons in the intermembrane space and to allow that proton to come back into matrix and make ATP in that process. So that's very straightforward. This is an interesting question, and this is from discussion section quiz 10, I think, photosynthesis, but the question is different. The figure is from that. But I'm asking you, what are the 3 conclusions you can draw from this figure? You may have never seen this, but I'm glad 67% got it correct. So simply understanding the x-axis is the leaf temperature, y-axis is the photosynthetic rate. So see, if you want to go through one by one, the bottom worst is a CAM plants. CAM plants have a very limited temperature tolerance, and they are very slow in photosynthetic rate, okay, or low in photosynthetic rate. Then come C3 plants have a the widest temperature tolerance from 5 degrees to 40 degrees, and they are medium in photosynthetic efficiency. And C4 plants can tolerate heat from 30 to 45 degrees Celsius, and they have the highest photosynthetic rate of all these 3 plants. Very simple drawing, but you're able to interpret that, okay? But still, you have a 26% didn't do it. Either you know this or you don't know this. So this is like a bimodal distribution of answers, okay? Then we're almost getting to the end. Briefly describe how light reactions and column are interconnected. And this is also, if you go back to the multiple choice question, you have the answers. We just looked at that, right? So this is what the inputs of Calvin cycle are, the outputs of light reactions. Okay, so that is, if you know these answers, and you should be able to 81%, more than 80% answered these correctly. And same here. So once you know that, this is not difficult to answer. Okay, so that is why use the multiple choice question as a word bank for you to jog your memory to answer this. Okay, this is a interesting, fun question. I know you must have seen this in your middle school or high school, but look at the classic experiment. That's the first discovery of oxygen in the atmosphere. Joseph Priestley's experiment of a bell jar with a mouse and a candle. It dies because the candle takes away the oxygen and there's no other oxygen supply. Whereas in this case, the candle is burning and the plants are releasing oxygen and they are consuming carbon dioxide from the candle. So the mouse lives longer, and 67% got it right. This is very similar to the previous question. You know it or you don't know it. So learn to understand, organize, remember, recall, connect the dots, see the big picture, and know the bottom line. I'm repeating this, it's like a parrot, but this is what is going to help you succeed in not only this class, but many science courses like this. Okay, I'm done. And I'm going to open up for questions.